Hello friends, good afternoon and welcome to EduSat Live Lectures. Dear friends, today in molecular biology we will be talking about regulatory RNAs. To discuss this topic we have with us our subject expert Dr. Charu Dogra Rawat. Dr. Rawat is Assistant Professor in Department of Zoology in Ramjas College, University of Delhi. Without further ado, I would like to welcome ma'am to our studios and request her to start the lecture. Welcome ma'am.
Thank you. Good afternoon students. So we have already talked about regulation, gene regulation, and primarily the regulation is carried out by proteins, the DNA which bind the DNA in sequence specific manners and either activate or repress the expression of the genes. But that can be done by the RNA as well. So today we will be talking about the role of RNA in regulating the expression of various genes. We will be primarily talking about the riboswitches and a phenomena termed as RNA interference, in which the gene silencing is done by the regulatory RNAs. So learning objectives particularly for this session would be to understand the diversity of riboswitches and their mechanisms of gene control, to understand the history behind the discovery of the phenomena RNA interference, to understand about the tools used in this phenomena RNA interference, and to understand the general mechanism of the RNA interference. So to start with, what are riboswitches? I would like to recall that we had talked about the tryptophan-operon regulation. The tryptophan-operon regulation, tryptophan synthesis enzymes are basically the genes for these enzymes are organized in an operon and they are transcribed as a polycistronic mRNA. The transcription regulation of tryptophan-operon was carried out by the RNA itself. There was a structure which was formed in the RNA which used to either abort the transcription or the entire 5 genes were used to be transcribed. So that was also the part of RNA which was regulating the one or the other structures it used to form, either an anti-terminator or terminator sequences. And we had already covered this in transcription regulation in prokaryotes. Riboswitches are a similar kind which are discovered in 2002 in bacteria as RNA-based intracellular sensors of vitamin derivatives. They are elements which are commonly found in the 5' untranslated region of mRNAs. So the riboswitches are the part of the mRNAs and are usually located as shown in the 5' untranslated regions. They exert their regulatory control over the transcript in a cis fashion, that is, they regulate the expression of the same RNA in which they are located, and they directly bind a small molecule ligand. Their regulation depends upon the binding of small molecules which are called as the ligands. And the directly metabolite binding to RNA causes riboswitching between alternate mRNA conformations that alter gene expression. So the basic mechanism by which riboswitches regulate the gene expression is by altering the conformation of the mRNA. That different conformations, morphological or structural features are distinguished and therefore they can alter, either switch on or switch off the gene expression of that particular mRNA which is there. So now the organization and mechanism of the typical riboswitch is as follows. The untranslated region, the 5' untranslated region has 2 domains. One is called as an aptamer domain and another is called as the expression platform. The aptamer domain is acting as a receptor that specifically binds a ligand. As I said, the small molecule binding is essential for them, for the riboswitches to exert their genetic control, and this aptamer domain is the part where the ligand binds. And the expression platform acts directly on gene expression through its ability to toggle between 2 different secondary structures in response to ligand binding. So ligand, when binds to the aptamer domain, the structural conformation of the expression platform changes, and which tells whether the gene is to be expressed or not. The common to both the domains, the aptamer and the expression platform, is the switching sequence, and its placement in the aptamer domain or the expression platform ultimately dictates the expression outcome of the mRNA. So, as it is shown here, when RNA polymerase is transcribing the mRNA, when the 5' untranslated region is transcribed, it attains a particular structural conformation. Upon binding of the ligand to the aptamer domain, the expression platform changes its structure organization, and therefore it can either form a terminator sequences referred to as by T or anti-terminator structure referred to as by AT. So, depending upon, like for example, in this particular case, without the binding of ligand, it exhibits an anti-terminator structure, and therefore the mRNA is transcribed fully. But when the ligand binds, the expression switches the decision, and therefore it attains the structural conformation of a terminator sequence, and then terminator structural conformation switches off the gene expression, and RNA polymerase dissociates, transcribing only this much of the mRNA. So, that is how the riboswitch work. The basic simplest class of riboswitches you can imagine are called as the thermosensors, RNA thermosensors. They basically sense the temperature. At low temperature, the helical structure is stable, resulting in the sequestration of the SD sequence, the Shine-Dalgarno sequence, by pairing with what is called as the anti-SD sequence, and this results in inhibition of the translation initiation. So, riboswitch alternate conformations can basically exert their role, exert their regulatory control by different mechanisms. They can inhibit the transcription, as it was shown in the previous diagram that the RNA polymerase was dissociating. They can also inhibit the translation, as these RNA thermosensors do. So, at low temperature, they attain a kind of configuration where the SD sequence is no longer available for the binding of the ribosomes, and therefore the translation is off, or the gene expression would not be there, as the protein will not be formed. At higher temperatures though, the ASD-SD helix is disrupted and the SD sequence is available for the translation initiation. So, the translation would be on, and the gene expression would be there, and the protein will be formed. No effector recognition domain is required, with the regulatory response instead tuned to the melting temperature of the inhibitory helix. So, for this RNA thermosensors, there is no metabolite or there is no ligand which is required, but only the change in the temperature is resulting in the alternative conformation of the mRNA, resulting in the translation to be on or be off. The classes of metabolites which binds to these riboswitch RNAs vary, and it is given there that the most common ligands belong to the, as they were when they were discovered by the vitamin derivatives such as flavin mononucleotide or FMN. The thymine pyrophosphate is found in eukaryotes, and then there are others such as S-adenosyl methionine, SAM and SAH, and then there can be amino acids which can act as the metabolites such as lysine and glycine. Then there can be purines which can which can act as the ligands for the riboswitches such as guanine and adenines. Then there are certain other ligands or metabolites which are also which can also act on the riboswitches such as the cyclic di-GMP or glucosamine 6-phosphate. And in fact, the ions such as magnesium can also act as the ligand. So, they vary from, there is a whole category of small molecules or ligands which can act on the riboswitches and therefore alter the gene expression. Now, we will look first about the mechanism of gene control in case of bacteria and the variety of riboswitches which are present. Now, this particular diagram shows on the top panel the different molecules which can act, or different metabolites which can act as the ligand. So, there are atoms which can act as the metabolites. Then there are metals such as magnesium 2+, there are purines and their derivatives such as adenine, guanine, ATP, etc. Then there are cofactors and other derivatives such as SAM and FMN, etc. And then there are amino acids such as lysine, glycine and other amino acids which can be basically act as the metabolites for the riboswitches. So, in the center you have a riboswitch where it is shown that there is a sensor and there is an expression platform. So, sensor is basically the aptamer domain. The boxes in the red basically shows that how they repress the gene expression, and the boxes in the green are basically for the activation of the gene expression. So, the mechanisms by which they do is the inhibition is done by the transcription termination, as I was already explaining that the binding of the ligand creates an alternate secondary structures, and the terminator structure is formed, and RNA polymerase is no longer able to cause the gene expression and therefore or transcribe the mRNA, and therefore the transcription termination occurs. This termination can be both by the row dependent or row independent manner. So, in the lower left panel it is shown as a row dependent terminator, where if you remember, then a row protein used to bind and cause the termination of the transcription. Then the translation can also be inhibited on the top right hand panel, where again there is a sequestration of the SD sequence, and upon binding the ligand, this SD sequence is no longer available, or Shine-Delgado sequence is no longer available for the binding of the ribosome, and therefore translation is inhibited. As far as the activation is concerned, the unbound RNA can be forming a structure called as the terminator structure, but upon the binding of the ligand, an anti-terminator structure can be formed, and this anti-terminator structure would let the RNA polymerase to read through, and therefore the transcription will be there. Then on the right hand bottom panel, there is a translation activation, that upon binding of the ligand, the sequester Shine-Delgado sequence is relieved, and therefore the ribosomes can now bind and cause the expression of the downstream target gene. So, by any of these mechanisms, the molecules at the top, upon binding to their particular riboswitches, can exert the gene expression regulation. So, in different cases, different things can happen. Then there are certain sugars, in for example gram-positive bacteria, the bound GLC-N6P induces cleavage by the GLMS riboswitch ribozyme in the 5' untranslated region. So, one of the mechanism by which this particular metabolite controls the gene expression is by the mRNA degradation and ribozyme after the ribozyme cleavage. So, ribozymes are the enzyme catalytic RNAs. So, upon binding of the ligand, the catalytic RNA will cleave the upstream untranslated region, and therefore the 5' OH of the resulting fragment stimulates the degradation of the message by the RNase J. So, cleaving by the ribozyme will let the RNase J to be bound to it, and therefore it will cleave the mRNA or degrade the mRNA which is there. Another mechanism by which the CDI-GMP regulates the gene control in, for example, Clostridium difficile is also shown. They do so by doing the alternative self-splicing and translation activation. So, C. difficile exploits an allosteric ribozyme riboswitch that combines self-splicing and translation activation. In the absence of CDI-GMP, the intron uses GTP cleavage site 2, thus yielding RNAs with truncated SDs that are not expressed. So, in the default state, the gene expression is off, but upon binding to this particular metabolite, binding of CDI-GMP to the riboswitch in the presence of GTP promotes the cleavage at a different site, which is say site 1. And self-excision of the group 1 self-splicing intron using splicing sites shown in green circles. This self-cleavage brings together two halves of the SD, and it becomes a functional Shine-Delgado sequence, and therefore the ORF is translated or the gene expression is switched on. So, in this manner, the CDI-GMP regulates the gene expression by alternative self-splicing and translation activation. Another mechanism which is found in bacteria is exhibited by the binding of SAM cofactor to the riboswitches. For example, in Listeria monocytogenes, the 3A and 3B riboswitches form anti-terminator hairpins and allow normal transcription in the absence of SAM. So, the default state, the gene expression is on. Binding of the SAM, however, causes transcription termination. The resulting mRNA fragments base pairs with the 5' untranslated region of the mRNA and functions in trans as an antisense siRNA that destabilizes the target RNA transcript, thus reducing the protein production. So, this mechanism is termed as cis transcription termination and trans translation repression. So, cis transcription termination occurs in a normal manner, that there is a anti-terminator sequence, terminator sequence which is a terminator structure which is formed upon binding of the SAM, but this small RNA which is now terminated can bind to the target mRNAs and in trans inhibit their translation or repress their translation. So, that is how the SAM exhibits its regulatory control. In eukaryotes, there is a TPP dependent riboswitches which are present, the thymine pyrophosphate which is present, and they do so by different mechanisms which are shown. For example, in the first one, they exhibit upon binding of TPP, there is an alternative splicing and there is a micro ORF translation which is there. It is found in fungi. The complementary base pairing results in preferential use of the distal set of splicing sites and elimination of the region between black dashed arrow lines. The resulting mRNA is translated to yield the full length product. However, the TPP binding to the riboswitch stabilizes the riboswitch fold, precludes the complementary base pairing and opens different sets of splicing sites that are otherwise sequestered. So, alteration in the configuration of this or the conformation of the riboswitch releases these particular sequences, the splicing sites which are otherwise sequestered. Splicing at the alternative splice sites removes sequences between the green arrow sites. The resulting mRNAs retain the micro ORFs which preclude translation of the main ORF located downstream of the micro ORF. Therefore, it is the main ORF expression is repressed. Another mechanism is found in algae in which the mRNA splicing in the absence of TPP eliminates a stop codon located within a riboswitch sequence. TPP binding, however, promotes again alternative splicing event that introduce a premature termination codon and disrupts the translation of the ORF as it is shown. In higher plants, sequestration of splice sites in the absence of TPP causes retention of the mRNA processing site or polyadenylation signal and yields stable mRNA with a short 3' untranslated region. However, upon binding of the TPP, the riboswitch sensor exposes the 5' splice site, causing the removal of the fragment between the green arrows containing the polyadenylation signal. The resulting mRNA now contains a long 3' untranslated region which is basically unstable and therefore it compromises the protein production. So, upon binding of this particular metabolite TPP in plants, in fungi and in algae, different mechanisms are followed to control the gene expression of the target genes. Another set of control which I should mention here is about the T-box system. In T-box system, when a high proportion of a tRNA with a specific anticodon is charged with the cognate amino acid, the tRNA anticodon loop can interact with the matching codon, which is called as the specifier sequence in an internal loop in the RNA. So, again how the RNA gene expression is regulated is basically when there is a high proportion of tRNA with a specific anticodon is charged with the cognate amino acid. So, when there is a high tRNA charging, it binds to a site at the riboswitch which is called as the specifier sequence, and usually this system is called as a T-box system. In the internal loop of the RNA, the presence of the amino acid at the 3 end of the tRNA blocks its interaction with the anti-terminator element. This kind of conformation is basically a terminator conformation and therefore the transcription is terminated. However, when there is a low tRNA charging, there are more free tRNAs available, that is which are not bound to their cognate amino acids, then the anticodon site binds to the specifier sequence and the site where the amino acid is no longer bound can bind to the anti-terminator sequences, creating a kind of a conformation which is an anti-terminator structure. The RNA polymerase can read through such a structure and therefore the gene expression or the transcription occurs. So, this kind of a system is termed as the T-box system, which is dependent upon the charging or uncharging of the tRNA. Each gene in the T-box family responds specifically to the cognate tRNA, with specificity determined primarily by the specifier sequence anticodon pairing. Discrimination between charged and uncharged tRNA occurs at the anti-terminator region. So, which genes or downstream genes are targeted depends upon the specifier sequences. However, whether the tRNA, there is a high level of tRNA charging or low level of tRNA charging is sensed by the presence of the amino acid which is there. So, that is all about the riboswitches, how these riboswitches regulate the gene expression. Next, I move on to talk about an important phenomena which was discovered called as the RNA interference. Looking at the history of RNAi, in 1989, Richard Jogrenson was working on petunias and he added the additional copy of the pigment gene under control of a strong promoter and expected that there will be a darker petunia which will be obtained. So, he was adding an additional pigment gene to produce so that the more protein for this pigment is more pigment protein is produced and therefore the petunia will be of darker in color. However, what he observed was not a darker pigmented flower, but rather such a kind of flower where there were which were termed as variegated petunias, in which there were certain petals which were completely white in color, and in fact some of the petunias obtained were completely white in color. So, actually he got plants with varying degrees of paler flowers, many that were sectored with purple and white regions, the variegated flowers, and even some of them were completely white. Now, this was very unexpected or unpredicted, because how can the increase in the number of the genes for a particular protein is resulting in the loss of its phenotype. So, a protein which is expressing a pigment color should ideally, if the gene number is increased for that pigment producing and specifically under a strong promoter, it will produce more of the pigment. But the result obtained were quite opposite to it, that actually the pigment was reduced because there were white colors, petals or white color flowers which were obtained. So, this basically perplexed the scientists for almost a decade, and in fact what they found out that the amount of pigment enzymes miRNA was greatly reduced. So, if in a normal purple flower there was so much miRNA which was present, the miRNA was reduced, which was quite obvious because there was less of protein and therefore thus there was less of miRNA also which was obtained. So, this particular question, as I said, was very confusing for the scientists, that how could introducing the extra copies of the gene reduce the amount of mRNA. So, for almost a decade this was unsolved, and finally the answer came from the worms. The Caenorhabditis elegans, the most common model organism which is used. The researchers were using antisense RNA to knock down the expression of PAR1 gene in worms. They could induce the phenotype predicted for the loss of PAR1 expression. So, when they started using antisense RNA, they could knock out this particular gene. They were trying to knock out this particular gene which is called as a PAR1 gene, and they expected that the introduction of the antisense RNA, because antisense RNA will go and bind to the PAR1 gene RNA, and therefore no protein production would be there, and therefore the phenotype of PAR1 would be lost. So, researchers were trying to do this. It was found that the sense RNA had the same effect. So, they used this sense RNA as a control. So, they were using antisense RNA, and antisense RNA would obviously exhibit the phenotype, and they did exhibited the phenotype that they could knock out the gene expression of the PAR1. But when they used sense RNA as a control, it was also producing the same effect, which was again astonishing. RNAs unrelated to the PAR1 gene did not had any effect. So, what was happening? Andrew Fire and Craig Mello demonstrated that it was in fact neither sense nor antisense RNA that silenced the gene. However, it was the double strand RNA produced by a mix of the two. So, they found that when they were using antisense RNA, it was actually contaminated by a little amount of sense RNA, and therefore these double stranded RNA molecules were actually causing the gene silencing. And similarly, when they were using the control in the sense RNA, there was a little bit of antisense RNA which was present, and this double stranded RNA molecules was causing the gene repression of the gene expression even in the control with the sense RNA. So, the antisense RNA and the sense RNA setups had produced a similar effect. This double-stranded RNAs were shown to be very potent in eliminating the expression of the target gene. So, later on, a lot of experiments were done to confirm and verify that these double-stranded RNA molecules can actually inhibit the gene expression of their complementary RNA. Now, the researchers then explained the petunia result based upon the observations in the worm, and they found that with clues from the worms, the scientists discovered that plants have a special RNA dependent RNA polymerase, which is called as RDRP, that produces double stranded RNA molecules when triggered by very high levels of mRNA. So, this made sense. Double stranded RNA had been shown to lower down the gene expression or cause gene silencing, and the scientists found that when the copy number of this particular pigment gene was increased, there was more number of mRNA was there, but higher level of mRNA triggered the RDRP to synthesize the antisense mRNA, and therefore double stranded RNA molecules were present in the petunias, which causes the repression of the gene expression for the pigment producing gene, resulting in the phenotype of the paler looking petals or complete white flowers. The gene silencing by double stranded RNA molecules has been termed RNA interference or RNAi. It is also called as the post-transcriptional gene silencing, and in fact it was such a revolutionary technique that in 2006 the Nobel Prize in Physiology or Medicine for their discovery of RNA interference, the gene silencing by double stranded RNA, was given to Andrew Z. Fire and Craig C. Mello in 2006. They received this Nobel Prize. So, now what are the tools for this RNAi? The double stranded RNA molecules we know, but that what are the different molecules which actually exhibit this gene silencing? So, there are three primary categories of RNA interference tools. One is called as the short interfering RNA, the microRNAs, and the pvRNAs or PV interacting RNAs or piRNAs which are there. The short interfering RNAs and microRNAs are the most broadly distributed in both phylogenetic and physiological terms and are characterized by the double-stranded nature of the precursors, as I was talking, we had already talked about, the phenomenon by which the RNA interference phenomena was discovered. pvRNAs or piRNAs are primarily found in animals, and they exert their functions most clearly in the germline and derive from precursors that are poorly understood but appear to be usually single-stranded. The siRNAs and microRNAs bind to the members of agoclad of the argonate proteins, as we will see down the lane. However, the PV interacting RNAs or piRNAs bind to the members of the PV clade. We will concentrate or focus on the siRNAs and microRNAs for today's discussion. So, initially the microRNAs and siRNAs appeared to be distinguished in two primary ways, and the ways were that microRNAs were viewed as endogenous and purposefully expressed products of an organism's own genome, whereas siRNAs were thought to be primarily exogenous in origin, derived directly from the virus, transposon or transgene trigger. So, the mRNA which is produced from the organism's own genome was processed to give rise to microRNAs, however, the RNA which was originating from anything outside the cell, derived directly from the virus or transposon or transgene, was termed as the siRNA or small interfering RNA. The second difference was that microRNAs appeared to be processed from stem loop precursors with incomplete double-stranded character, You know that RNA has a tendency to form secondary structures such as stem loop structures or hairpin structures. So, when a stem loop structure is formed, then there is an incomplete double stranded region that occurs, and microRNAs are derived from such structures, whereas siRNAs were found to be excised from long fully complementary double stranded RNA molecules. But one thing was clear, that both classes of these small RNAs were quickly revealed to depend upon the same two family of proteins. And the two family of proteins, the one is the dicer, the enzyme which excises them from their precursors, and another is called as the agoprotein to support their silencing effector functions. So, in all, the three sets of macromolecules, the dicers, the agos, and approximately 21 to 23 nucleotide duplex derived RNAs become recognized as the signature components of the RNA interference. The common aspects of all microRNA and siRNA pathways are shown. So, there is a double stranded RNA precursor which is there. The dicer is an enzyme which chops this double stranded DNA molecules into smaller molecules. These smaller molecules, which are either siRNA or microRNA, the processing is different, but we will talk about that in later in our sessions. But these smaller molecules will then associate with the agoprotein molecules to give rise to a complex which is called as RNA induced silencing complex or RISC complex. This RISC complex will then recognize the target mRNA which is shown. So, initially it is a double stranded RNA molecule which associates with the RISC complex. The passenger strand then leaves and the guide RNA remains with the RISC complex, and now it is said to be an activated RISC complex. This activated RISC complex then bind to the target mRNA and brings about gene silencing in any of the ways. It can inhibit transcription, it can increase heterochromatinization, it can inhibit translation, it can stimulate RNA degradation, and it can also stimulate DNA degradation. So, it can exert its control by any of these mechanisms. So, this is the common aspect of all miRNA and siRNA molecules. Looking at the dicer, which is a portal into RNA silencing, it is characterized by several domains in a specific order, such as DEX-D-H ATPase domain. It requires the energy of ATP to cause the cleavage. A PAS domain, an RNase 3 domain, and a double stranded RNA binding domain. The PAS and RNase 3 domains play central roles in excising the microRNA and siRNA preferentially from the ends of the double stranded RNA molecule. So, PAS domain basically holds it, and RNase 3 enzyme basically cleaves the target mRNA which is there. Then looking at the Argonaut protein, which is at the core of RNA silencing, is the Argonaut superfamily can be divided into three separate subgroups: the PV clade that binds the piRNAs, the ago clade that associates with microRNAs and siRNAs, and a third clade that has only been described so far in the nematodes. So, the ago clade associates with the microRNAs and the siRNAs, and these proteins are defined by the presence of four domains: the PAS domain, which shared with the dicer enzymes, the PV domain that is unique to the Argonaut superfamily, and the N and the mid domains. The 5' end of the guide strand associates with the binding pocket in the mid domain, and the 3' end binds to the PAS domain as it is shown. The remainder of the guide tracks along a positively charged surface to which each of the domain contributes. The target cleavage site is juxtaposed with active site residues in the PV domain. So, Argonaut also causes the cleavage of the target mRNA which is there. So, these are the general history about the RNA interference and general mechanism on how these small RNA molecules, basically the microRNAs and siRNAs, regulates the gene control. this session, we will talk about how the RNA interference tools, the small interfering RNA and the microRNA exhibit their control over the gene expression. So, particular learning objectives would be to understand the mechanism of small interfering RNA based gene silencing, to understand the biogenesis of microRNA in its assembly into miRISC complex, to understand the mechanism of miRNA based gene silencing, and to understand or basically look a little deeper into the evolution and exploitation of this RNA interference phenomena. So, these siRNA sources are basically derived from, from a variety of sources which are there. They can be derived basically, siRNAs were originally observed during transgene and virus induced silencing in plants consistent with the natural role in genome defense. In 2002 and 2003, centromeres, transposons and other repetitive sequences were uncovered as another wellspring of siRNAs. The trans-acting siRNAs, also called as tacrNAs, that are diced from specific genomic transcripts and regulate discrete sets of target genes. So, initially siRNA and microRNA were distinguished on the fact that microRNA are usually derived from its own genome. However, the siRNAs were mostly from the exogenous genome such as the viruses or the transposon or other genomes or transgenes which are there. So, siRNAs used to be derived from that. But as you can see, there are diversity of siRNA sources which are there, and subsequently it was found that in fact the own genome such as the centromeric regions can also give rise to, or the tacrNAs in which the trans-acting siRNAs are diced from the specific genomic transcripts which are basically present there could be found. Other sources of endogenous siRNAs that have been identified include convergent mRNA transcripts and other natural sense-antisense paired duplexes involving pseudogene derived antisense transcript and the sense mRNAs from their cognate genes and the hairpin RNAs which are there. So, there are a whole diverse sources by which the siRNAs can be derived. So, again looking at the mechanism of this siRNA silencing, the double-stranded DNA molecules, the siRNAs which are derived from the precursor by the dicer enzymes are now associate with the agoproteins. So, the agoprotein initially associates with the complete double-stranded molecule and then subsequently the passenger strand is lost and degraded and it becomes an activated RISC complex. The guide strand gets associated and will take it to the complementary, it will base pair with the target sequence by the complementarity, Watson-Crick complementarity base pairing, and then it will cause the gene silencing. The siRNA amplification by RDRP is also there, which can regenerate the siRNAs and add it to the siRNA pool. So, RNA dependent RNA polymerase can synthesize the complementary strand for the small RNA molecule, and it can again become it can be added to the double stranded siRNA pool which is there. Now, the gene silencing can be occurred by different mechanisms. So, you can see in the center there is an activated RISC complex which is there, and if there is a partial and usually there is a complementary mRNA which is very much complementary, 100% perfect complementarity to the siRNA, it will bind to that target mRNA, and agonode, which is also called as a slicer, the agoprotein will cleave the mRNA which is shown on the right hand side lower panel and will cleave the target mRNA which is there. If there is a partial complementarity, it can also work as an miRNA based mechanism. If there is a partial complementarity which is there, it can repress the gene expression by causing translation inhibition or by causing exonucleotic degradation which is there. Apart from that, another mechanism by which siRNA causes gene silencing is by inducing heterochromatinization. So, there are certain markers for heterochromatinization such as methyl transferases HMT, which will methylate or demethylate the certain regions on the DNA, or there is a histone methyl transferases which are there, which will add on methyl groups to the certain positions such as lysine 9 position on the histones in the nucleosomes. So, by adding these repressor markers, it can cause the heterochromatinization and therefore inactivation of the gene expression. This heterochromatinization gene silencing is particularly studied in case of Saccharomyces S. pombe, and where the silencing of centromeres occur and results in the switching of the mating type. So, a model for the RITS recruitment and silencing of centromeres in S. pombe is there, where transcription is terminated. So, RITS basically stand for RNA induced transcription silencing. So, as you can see on the top panel, the repeat sequences are transcribed from both strands by RNA polymerase II, generating double stranded RNA, that is substrate for the dicer. So, there is a typical nucleosome spaced arrangement on the topmost part, and there is a particular sequence, the centromeres repeat sequence lying in between. This sequence is transcribed from both the ends, resulting in the double stranded RNA. These double stranded RNA is then chopped into smaller portions by the enzyme called as dicer. Then this small RNA, interfering RNAs or siRNAs, will associate with the complex called as the RNA induced transcription silencing complex. They associate with these proteins, and siRNAs thereby produced are loaded into the Argonaute containing complex, the RNA induced transcription silencing complex. The siRNA containing RITS is then recruited to the Pol2 tethered transcripts being generated by continued transcription of the centromeric repeats through complementarity between the siRNA and the transcript. So, siRNA now has a perfect complementarity with the transcript of the centromeric region. So, as their centromeric region is transcribed, this RITS complex along with the siRNA will complementarily bind to this particular RNA. The complex then recruits certain other factors such as CLR4 and SY6 that locally modify the nucleosomes by adding the H3K9 silencing markers. So, there is a lysine 9 position, basically the histone modification occurs and the methyl groups are added, and therefore the silencing is occurring. The slicing of the transcripts by Argonaute within RITS generates substrate RNAs for the RDRP or the RNA dependent RNA polymerase, which synthesizes a complementary strand and thus generates further substrate for dicer. This process is required for the nucleosome modification and thus the region silence to be spread. So, centromeric region silencing is spread throughout by slicing of the siRNA producing and RDRP, then synthesizing the antisense RNA and creating a double stranded RNA molecule which can then be cleaved by the dicer and therefore follow the or spread this heterochromatinization which is there. So, gene silencing basically can occur by any of the mechanisms, that is the translation inhibition, the exonucleotic degradation, although most common form of siRNA gene silencing is by mRNA degradation which is there, and sometime in some organisms such as in yeast pombe it can also introduce heterochromatinization which is there. Now, we will look at the biogenesis of microRNAs, the another RNAi tool. The two specialized now, microRNA has an extra step before the dicer enzyme which is there. Usually it is, as it was said, that usually it is produced by the endogenous gene transcripts. So, the genes are transcribed in the nucleus and the mRNA are produced. The long primary transcripts are then processed by different enzymes, usually two sets of enzymes are required to process the initial mRNA transcript containing the stem loop structure into the mature microRNA. So, this is how basically the stem loop structure of a miRNA transcript looks like. So, it is a stem loop structure and then there is a lower stem which is approximately 11 base pairs, there is an upper stem which is a 22 base pairs long, and then there is a terminal loop which is there. Now, you can see the red arrows indicate the cleavage cleavage by an enzyme called as drosha. So, the processing by this enzyme is additional for the processing of the siRNA which is there, and the blue marker shows the cleavage by the enzyme which it shares with the siRNA formation which is called as the dicer. So, drosha, which is a nuclear member of the RNase 3 family, with the aid of other proteins called as DGCR8, or it is also called as PASHA in some, cleaves this primary miRNA transcript to generate a pre-miRNA species. Drosha makes two cleavages as it is shown. The pre-mRNAs can be encoded by any part of a transcript coding region, leader region or introns. It can be basically coded by anywhere. The DGCR8 contains two double-stranded RBD domains and stably associates with the ribonuclease to form what is called as the microprocessor complex. Drosha, as I said, resides in the cell's nucleus, and the drosha catalyzed cleavage event occurs in this cellular component. So, now describing a little detail about the recognition and cleavage of the pri-miRNA by the microprocessor complex. So, you can see there is a pri-miRNA which has two stem regions which are distinguished. One is an 11 base pair region and a 22 base pair region which is there, which is approximately 33 base pair. The total pri-miRNA, the 3 helical turns of double stranded RNA molecules are there. The drosha cleaves 11 base pairs away from the double-stranded RNA single-stranded junctions, that is between the lower and upper stems of the pri-miRNA. So, you can see the DGCR8 or PASHA is associated with this double-stranded region, and then drosha comes and causes the cleavage at the 11 nucleotide stem, after the 11 nucleotide stem region. The two cleavages thus generate the approximately 65 nucleotide pre-microRNA composed of 22 base pairs or 2 helical strands. The RNase 3 family enzymes are specific for double-stranded RNA and cleave it in a manner that leaves a 2 nucleotide overhang in the 3' end of the double-stranded RNA product. This 3' overhang is important for the recognition of that RNA molecule by the next enzyme in the pathway dicer. So, the biogenesis of microRNA requires this extra step in which the cleavage by drosha aided by different proteins such as DGCR8 occurs. This occurs inside the nucleus, and the pre-microRNA which is generated is then exported out of the nucleus into the cytoplasm, where the dicer enzyme is located, and then dicer process these particular microRNAs which are generated to form the functional microRNAs which are there. So, as you can see that the pre-microRNAs are exported out where they associate with other aiding proteins such as TRBP, and dicer also associate, which recognizes this pre-microRNA by a 3' overhang, a 2 nucleotide 3' overhang, and then it cleaves them into the smaller microRNA molecule. These microRNA molecule then associates with the RISC complex, in this case termed as a miRISC complex, where then the miRISC complex activates, the passenger strands usually leaves or degraded, and the activated or the guide strand then guides this miRISC complex to the target genes which are there. So, pre-microRNA is exported from the nucleus and processed by dicer to form the mature microRNA or microRNA passenger strand duplex. After processing, the microRNAs are assembled into microRISC. miRISC, only one strand of the duplex is stably associated with an miRISC complex, and the other strand, which is basically the passenger strand, also gets associated with the miRISC complex, but it is not stable. However, the miRNA strand is usually more strongly favored than the miRISC strand which is there. So, in this way they assemble into this miRISC complex and then they mediate the repression. Now, the repression basically, or the gene down regulation of the gene expression exhibited by microRNAs is at the level of translation. It also exhibits at the level of translation, such as it is shown in this particular diagram. So, in normal condition, which is shown at the topmost portion, the non-repressed mRNAs recruit initiation factors and ribosomal subunits and form circularized structures that enhance translation. So, normally this particular mRNA transcript will be translated into protein which is there, but when miRISCs bind to mRNAs, they can repress the initiation at the cap recognition stage. So, you know that ribosomes bind by recognizing the cap. So, they compete for the cap binding. So, they do not let the methylation cap to be present at the 5' end of the transcript which is there. So, they do not, and therefore, they repress the initiation of the translation. They can do so also by the 60S at the 60S recruitment stage, where they compete for the elongation factor B or 60S stage. So, they would not let the 60S component to bind to the transcript and therefore cause the translation repression, and therefore causes a translation repression. They can also induce the deadenylation of the mRNA and thereby inhibit the circularization of the mRNA which is there. So, the mRNA transcript has to circularize for the translation to occur, but they induce deadenylation of the mRNA. So, there is no more polyadenylation tail which is required, is no more present, and therefore, this inhibits the circularization of the mRNA and therefore inhibits the translation. Another mechanism by which it can do is it can repress a post-initiation stage of translation by inducing ribosomes to drop off prematurely. They can also promote the mRNA degradation by inducing deadenylation followed by decapping. So, mRNA which is not having a cap or which is not having a polyadenylation tail will be degraded by the degradation RNA degrading machinery. So, they can also cause the decapping and the depolyadenylation leading to the degradation of the mRNA. So, by any of these mechanisms, the microRNAs can actually repress the translation of the proteins and therefore controls the regulation of the gene expression or regulates the gene expression which is there. So, this is how basically the small interfering RNAs and microRNAs exhibit their repression. Looking at the evolution of microRNAs, that why did the microRNA ever evolve? Because RNA interference machinery was present in the cells, but for example in the case of yeast, the RNA interference machinery is present, but microRNAs are not found. So, they said that one ancient function of the RNAi machinery might have served and still serves is protecting organisms from the transposon and viruses. So, why this RNA interference machinery was actually present in the cell, that it acts as an immune system. It protects the own genome by the invading genomes by repressing the gene expression, by creating the double-stranded RNA molecules from the foreign genes or transgenes or virus or transposon bringing genes which could be lethal to the cell, and therefore suppresses its gene expression which is there. In humans, for example, the miRNAs can act as tumor suppressors of the oncogenes which is there. For example, there are certain microRNAs, the low expression of these tumor suppressor microRNAs are associated with the inhibition of the oncogene mRNA which is there. So, miRNA that normally downregulates an oncogene can function as a tumor repressor gene. The loss of function of the microRNA by mutation or deletion might result in an abnormal expression of the target oncogene, which would then contribute to the tumor formation. So, why did microRNAs evolve? So, one of the reasons could be that, for example, in case of humans, they act as the tumor suppressors of the oncogenes or the cancer producing genes which are there. Then some of the genes, the higher expression of these microorganisms can induce actually the tumor formation. So, the amplification or overexpression of a microRNA that downregulates a tumor suppressor or other important genes involved in differentiation may contribute to the tumor formation by stimulating the proliferation. So, normally the low expression of this microRNA will inhibit the formation of the cancer which is there, but when the higher expression is found, then it might probably inhibit the differentiation and can lead to the activation of the genes which are required for the proliferation or pervasiveness or angiogenesis and all those cancer related things, and therefore induce cancer which is there. Then RNAi has also become a powerful tool for manipulating the gene expression. Since its discovery, it has been a very, very strong tool for manipulating the gene expression of different genes, and it can be used in this manner. It can be induced in worms by feeding the bacteria expressing the double-stranded RNA molecules. Any desired double-stranded RNA can be expressed in the E. coli on which the worms feed, and when they feed on this E. coli, these double-stranded RNA molecules will enter the gut of the worms and basically into the cells, and from there it will deliver the enough substrate for the RNA interference response to be triggered, and basically you can knock down or shut down or silence any gene of the worm by this particular mechanism. So it was exploited. To know function of the gene, the best technique is to knock it off, and therefore the ones which phenotype you can assay, then you can know that functional association is also there. But for a sustainable expression of double-stranded RNA molecules, instead of trying to deliver these double-stranded RNA into cells, the investigators found it more fruitful to mimic microRNAs. To this end, libraries have been generated in which short genes are synthesized as oligonucleotides and cloned into plasmids. Each short gene is designed to give a transcript that will fold into a stem loop inside the cell. These are processed by dicer in the cell to generate an siRNA that will direct silencing of its target genes, as it is shown in the diagram that a PCR amplified product is cloned into a vector, and this vector is introduced into the E. coli. Now the E. coli, it does not get expressed by the E. coli DNA polymerase, but is exhibited by a T7 RNA polymerase which is present there. These short synthetic genes are called short hairpin RNA genes. By using an appropriately designed shRNA, any individual gene in the genome can be targeted. So basically this RNA interference is an important genetic manipulation tool used in the biotechnological industry, where any gene can be knocked off or shut down, where one of the gene is exhibiting a particular phenotype, and then that phenotype can be assayed or studied, and in fact a whole library of plasmids carrying different shRNAs can be created. That introduction can actually inhibit each gene of a particular organism which is there. So RNA interference phenomena is basically a very important phenomena, and a lot of regulation is done by the RNA, and we discussed about the riboswitches and the RNA interference or the microRNAs and siRNAs which regulates the gene expression in a particular cell.
Okay, on that note, I would like to thank ma'am for this very interesting discussion, and I hope, my dear friends, that you have been hugely benefited by this lecture and by what regulatory RNAs are all about. On that note, I would like to thank you, dear friends, for watching our show. Stay tuned and keep watching. Thank you.